Good day, dear students. This video is about phototransduction, the phenomenon by which photons or units of light energy is converted into visual image in the brain. This is competency 10.17 within the framework of the new competency-driven curriculum, which is physiology of vision. The learning objectives are to explain the similarities and differences between the eye and the camera, list the photoreceptor cells, and list the layers of the retina, and describe the mechanism of phototransduction, which is the main part of this video. Introduction to the eye. It is widely and popularly believed that the eyes resemble a camera. Now I'm going to give you an activity. You can think for a couple of minutes, pause this video, and compare the similarities and differences between the eye and camera. If you agree, give reasons for your answer. If you disagree, give reasons for your answer. You can give two reasons for yes and no. Like the camera, the eye has a lens system. It has a variable aperture system, the pupil, and it has a film. The film is the retina. But here the similarity ends. Now, if we take this belief at face value that an inverted image is formed on the retina and that image is converted into a straight image in the brain, then see what is happening. To see this image in the brain, there would have to be a still smaller human being to see the visual image in the brain. This is a Homounculus. And to see the image in this man's brain, there would have to be a still another smaller person. So there would have to be an infinite number of images, eyes, and humans to see this image. This is known as the Homounculus fallacy. What I want to explain and emphasize is that the brain sees and not the eyes. The visual image is formed in the brain. Therefore, we see with the brain and not with the eyes. See another example. A man with vision can see a chair. But if you describe a chair to a man who has lost sight recently or who is blindfolded, then he can still perceive the image of a chair once a chair is described. So this means that the image is formed in the brain. This is a man called Mike May. He lost his vision at 3 due to an explosion, as a result of which his cornea scared. Then he got a stem cell transplant and got his cornea repaired. The whole media was there to watch him. But see what Mike said. My brain is going, oh my gosh. So Mike couldn't recognize his children. He was not knowing what these flashes of light from the journalists were. When he was taken in the car, he could not recognize the traffic. He ducked under an overbridge, thinking that it would hit him while he was still in the car. He couldn't recognize the stop sign. And above all, he couldn't ski anymore, which he was doing before he regained his vision. So with these three examples, we can understand that we see with the brain and not with the eyes. We see with the brain and not with the eyes. So image formation, retina forms electrical signals. And these electrical signals are converted into an image in the visual cortex. And this image is what we see. So perception occurs at the level of the brain. A perception of reality has more to do with what's happening inside the brain. To define phototransduction, it is the process by which a photon, a unit of light energy, is converted to electrical signals in the retina. Transduce means to lead across. Now, an overview of the neural organization of the retina. These are the rods and cones. They are the photoreceptor cells, and leading from the rods and cones, there are two cells, bipolar cells and ganglion cells, which lead to the optic nerve. So there is from the retina, there is a 3-neuron pathway to the optic nerve. The photoreceptor cells, that is the rods and cones, the bipolar cells, and the ganglion cells. And then there are two cells which mediate lateral inhibition, the amacrine cells and the horizontal cells. So photoreceptor cells, rods and cones, bipolar cells, ganglion cells, and layer of optic nerve fibers. So there are 5 major neurons in the retina: rods and cones, bipolar cells, ganglion cells, and then amacrine cells and horizontal cells. These are the layers of the retina. Now for describing these layers, there is a mnemonic. In my own garden, I put in new and old plants now with extra pesticide easily. That is the inner limiting membrane, optic nerve layer, ganglion cell layer, inner and outer plexiform layer, inner and outer nuclear layer, external limiting membrane, photoreceptor layer, and epithelium. When you are in dark, the rods are responsible for vision in the dark. Rods are extremely sensitive to light. They are receptors for night vision, and this is known as scotopic vision, which is in the antelopes and animals in the jungle who have just two-colored vision. They detect the details and boundaries of the objects, which is important for them to detect a predator or find a prey. In light, cones have a greater visual acuity. Cones are able to see in light. Cones are responsible for vision in bright light. This is known as photopic vision. Now the mechanism of phototransduction. Imagine you are on an African safari, and you are on a safari in a dark night, and some meters away from you is a lion. So as one moves to a dimly lighted environment, the retinas become slowly more sensitive to light, and the individual becomes accustomed to the dark. This takes place over 20 minutes. So this decline in visual threshold is called as dark adaptation. Now the rods have become active, and this is known as scotopic vision. When you go to the safari in the morning, when you go in a vehicle, there is light adaptation. The visual threshold decreases. The cones have become active, and this is known as photopic vision. In a moonlit night, both rods and cones are active, and there is mesotopic vision. How does phototransduction occur? First, let's understand the structure of the photoreceptor cell. The photoreceptor cell has an outer segment, an inner segment, a nucleus, and a synaptic body. This outer segment has the photopigment rhodopsin, and 40% of it is made of rhodopsin. Normally in the dark, there is less release of neurotransmitter glutamate. This is because what happens is that this sodium potassium ATPase pump is pumping out sodium ions, and these sodium ions are transported back to the outer segment by cycling GMP gated channels. They are also transported to the synaptic body, and so the Receptor potentials in the photoreceptor cells is minus 40 millivolts, which is much less negative compared to the minus 70 millivolts in the other sensory receptors. Cyclic GMP keeps the sodium channels in an open state. How does phototransduction occur? When light strikes the retina, the retinal pigment rhodopsin is activated, and this activates a G protein. What is this G protein called as? It is called as transducin. This G protein activates an enzyme called phosphodiesterase. Just as other G proteins activate the enzyme adenyl membrane-bound enzyme adenyl cyclase or phospholipase C, this G protein transducin activates a membrane-bound enzyme called phosphodiesterase, which breaks down cyclic GMP. This is the main step, breaking down of cyclic GMP. Once the cyclic GMP breaks down, the cyclic GMP gated channels close and the receptor potential becomes more negative. That is, it hyperpolarizes. This is the only sensory receptor in which the firing of the electrical signal occurs when the receptor potential becomes more negative. There is hyperpolarization. So the mechanism of phototransduction: incident light, structural changes in the retinal of the photopigment, conformation changes in the receptor protein rhodopsin, which activates phosphodiesterase, which decreases intracellular cyclic GMP, which results in closure of sodium channels, and which results in hyperpolarization and decreased release of neurotransmitter glutamate. And as a result, responses generated in the bipolar cells and other neural elements. Please remember, in the bipolar cells, up to the bipolar cell levels, all or none red potentials are only generated, but all or none action potentials are generated only in the ganglion cells. And the rods fire when there is increased release of neurotransmitter glutamate, when the receptor potential comes back to its normal level. So the cones are responsible for vision in light, that is photopic vision. They are responsible for color vision, and the rods are responsible for vision in the dark, that is photopic vision. And when there is both light and dark, there is mesotopic vision. Both rods and cones are active. So thank you and have a good day.